let's keep this going, guys. Our next speaker is here. He's backstage. Let's bring him on. Dan Alkon. He is the president and chief scientific officer. There he is. See, I said it and it happened. It's like magic. Chief scientific officer, ticker SNPX, Synaptogenix. Dan, welcome to the Benzinga Small Cap Healthcare Conference.
Thank you, Spencer.
I'm going to get off the screen here and I'll move backstage, and then the floor is yours.
Thank you so much. So good afternoon. I'm here to talk about a neuroregenerative, a breakthrough strategy to treat degeneration of the brain. And for those of you who are not familiar with the field, of course, Alzheimer's disease is the paradigmatic example of neurodegeneration. If we can really treat this underlying disease, we're talking about tens of billions of dollars. If you add in the other types of degeneration, such as Parkinson's disease, multiple strokes, Fragile X mental retardation, traumatic brain injury, all of these involve degeneration of the brain and the loss of the wiring in the brain, the loss of the connections in the brain, which makes up that wiring. We have developed a strategy which we believe is a breakthrough strategy. We've pioneered it beginning 10, 12 years ago, first starting at the NIH, at the Neurologic Institute, and then at the Rockefeller Neuroscience Institute, where I was the founding scientific director. And we developed this strategy to actually reverse the wiring loss. The elephant in the room in this field, the reason why so many major companies, as well as small companies, have failed to actually treat the underlying disease and to actually win the approval of the FDA for treating the underlying disease. Not like just modestly slowing it down, such as Biogen offers for mild cognitive impairment. We're talking about actually reversing it, rebuilding the connections that have been lost. We've developed this strategy beginning 10, 15 years ago, first at the NIH and then at the Rockefeller, when we saw that memory storage, memory itself, involves laying down of more wiring. And we saw that there were certain drugs that could engage the synaptic growth factor pathway, such as BDNF, brain-derived nerve factor, or nerve growth factor. These are intrinsic to the brain. They're already there. And we have a way of engaging them and bringing them back to their former active self in the developmental stages right after we're born, where we're starting to lay down those networks and building up the brain connections. They tend to fade. And in Alzheimer's disease, they tend to fade even more. So one of the hallmarks of the Alzheimer brain is not only amyloid plaques and tangles, which are the familiar red flags that everyone knows about, but the actual loss of the synaptic growth factors that stimulate the laying down of new wiring. We found out a way, pharmacologically, with our lead compound, Bryostatin, and with other compounds in our platform, to actually stimulate and engage those synaptic growth factors, somewhat like an orchestra of synaptic growth factors that can be brought online. And for this reason, it's doing something that no other company, no other strategy has ever been able to do. And that is to actually restore the wiring, the brain wiring that's been lost, and restore the cognitive functions that have been lost. So just in a brief summary in this slide, you see some of the diseases that we could potentially address. Alzheimer's disease, for which we already have successful pilot phase 2 trials, multiple sclerosis, Fragile X, Parkinson's, traumatic brain injury. We plan to work in these spaces. We have an ongoing trial that's continuing the first 2 trials that we've done, which showed improvement above baseline, not just slowing down of the disease, but improvement above baseline in Alzheimer's disease. We now have an ongoing longer trial, NIH-sponsored, that's going to read out in the midsummer, third quarter, so early third quarter of next year. It's ongoing. It's more than three quarters enrolled, and it's totally safe and has been safe in our other 2 trials. In addition, we have announced a collaboration with Nemours DuPont Hospital, where we will be treating Fragile X children who are born basically with defective synaptic connections, and which we've shown in preclinical models that we can actually restore those connections. And in addition, restore the cognitive functions that they are mediating, the cognitive functions that are lost as a result of the loss of the wiring. In addition, other laboratories and other brains and medical centers have been piloting with our protocol, with our drug and our protocol, other diseases that involve degeneration, Parkinson's disease, multiple sclerosis, etc. The reason why the FDA has given us orphan drug status for our lead compound Bryostatin is because the Fragile X Foundation used our protocol as well and got very good results, very similar to ours. In addition, they looked at autistic spectrum disorder metrics and found that those two were tremendously benefited by our drug. So what we're talking about, again, is doing something that has not been done in the last 20 years, and that's the reason why this field has been so difficult, because they're looking at, in my opinion, the wrong targets. Amyloid plaques and neurofibrillary tangles are not correlated with cognitive deficits. Very loosely related, but not closely correlated. In fact, our old people know, and we know that when they go into their 90s, they're acutely functioning cognitively, and when they pass on and look at autopsy, their brains are riddled with amyloid plaques and neurofibrillary tangles, yet they're functioning perfectly well. When Clive Holmes in England obtained brain samples at autopsy from the Pfizer and J&J Alkon trial that was failed, he found that they got rid of the amyloid plaques, but the patients didn't get better. It should have been a watchword. It should have been to warn big companies that keep pursuing these targets with very modest efficacy. This is why the FDA has not approved any drug, including the Biogen drug, to actually treat the underlying disease. And that is what Synaptogenix is going after, treating the underlying Alzheimer's disease, dealing with the synaptic loss. The question is, what are those synapses? In the next slide, you have an idea in the next slide what they actually look like. In a dish on the left-hand side, these are synaptic networks, cultured neurons that we see in the laboratory, and they've been treated with toxic proteins from Alzheimer's disease patients. But if we treat that same dish with a drug, we can totally prevent and restore those connections. And this was shown in vivo in clinical models, in animal models, and now in clinical trials, we've seen actual improvement of patients over baseline. In 2 trials, which we hope will be published in great detail in the near future, where we'll actually show how these 2 trials reinforce each other, exactly the same conditions, and when pooled show tremendous benefit at the 001 level. So these are our objects, but it doesn't mean we're not also going after amyloid, because our same drug that promotes neuronal health and synaptic regeneration also promotes the elimination of amyloid and tau. It does all those things. So it has a multimodal efficacy that we think is great for Alzheimer's, but it's also great for other neurodegeneration objectives. And that's what I hope that you might take away from my brief talk this afternoon, is the idea of understanding that we're talking about neurodegeneration and neuroregeneration as a whole. That's why we are going after these other trials. That's why other medical centers using our protocols have obtained such good preclinical results as well. In the next slide, you can see that we can actually track the benefit. This is a compassionate use trial patient. In the right-hand side in that graphic, you'll see the green circles represent the mini-mental status exam of the patient who was very severe. He was sitting in his chair, hallucinating, staring at the ceiling. When we started treating him at those arrows at the bottom, on the axis at the bottom, each arrow representing a dose, he shot up. His mini-mental status exam went from about 2 or 3 all the way to 12. And as long as the target of our drug, Bryostatin, was elevated, as indicated by the blue circles, the patient was better. When the levels started going down. We let up on the dosing. The patient, including with the ADL severe, got worse. When it goes back up, he goes back up. So there was a close relationship between the target of Bryostatin in the human patient's bloodstream and the cognitive benefit. This is a biomarker of the actual cognitive benefit that we were viewing. The next slide, we see an example of one of our first trials. This was a short trial. It was a 15-week trial, and in the orange on the left-hand side, starting from 0, which is the baseline, in the absence of anything blocking the NMDA receptor, which is memantine, you see a very significant elevation of the severe impairment battery, the SIB. And it keeps on giving. It keeps on going up. This is what we need to treat this disease. We don't need something that's just going to last for a day or a week, a couple of weeks, but for months. In fact, you see that this keeps getting better, and even 30 or 40 days after all drug administration has ceased, it's still getting better. So we're looking at an improvement of the patient over 0, over his own baseline, that is sustained. It's a sustained benefit. And that sustained benefit, we think, is consistent with the kind of rebuilding of synaptic networks that we had seen in preclinical models. And you notice the two control groups, one light green and the blue at the bottom, you don't see any benefit at all, and actually a reduction, which is typical of the severe impairment battery, which is the metric of choice for advanced Alzheimer's disease patients, which is what cohort we are treating. We believe we're the only company that's actually focusing on advanced, not mild cognitive impairment, not very early Alzheimer's disease, which is extremely difficult to diagnose, even with very good clinical diagnosis, and even with PET scan. It's very inaccurate in those early weeks and months. But nevertheless, we believe that we can treat that as well. So in a kind of summary of the benefits we've already seen, we have a safety profile that's absolutely clean. There are no safety issues at all. We've seen cognitive improvement over baseline, not just a slowing down like 20 or 25%, as, for example, seen with Namenda when it was registered and approved in 2003, or with the Biogen drug when it was recently approved for mild cognitive impairment, about a 20%, 22% slowing. We're not talking about a slowing. We're talking about a reversing. We're talking about bringing the patient back. And we believe that with successive dosing cycles, we can keep bringing the patient back. And as I mentioned, even when the drug is no longer being administered, it's still effective at least 1 month after dosing. In the next slide, we look at a few of the goals. We showed that we cannot have standard of care Namenda being administered because it blocks one of the key targets of PKC epsilon, which is the synaptic growth maestro. It orchestrates the synaptic growth factors to rebuild the connections. We are grant supported, and now we are focusing on patients who are not the most severe, still very advanced, still quite advanced. But by doing this, we believe we streamline the trial and we reduce noise in the trial. And that's why we're very optimistic that we're going to see the kind of benefit that we hope to be describing in the literature shortly in a full-length peer-reviewed article that we hope will appear soon. In the next slide, you see a kind of very brief Gantt chart. We're out in phase 2 with Alzheimer's disease. We hope we're setting ourselves up for our phase 3. We believe that the FDA will be very favorable toward that step. And thereafter, right behind it, are Fragile X multiple sclerosis, for which patients we have lined up superb clinical center collaborators, one of which we've announced already, Namur DuPont, the other which we hope to announce very shortly. Next slide. Just to give you an idea of why we think in this tens of billions of dollar market potential indication and related indications, we are very undervalued. Our market cap is now about 50 or 60 million, but compared to what Athera or Cassava have in the essentially billion-dollar level, it's minuscule. And yet our data preclinically and even clinically, we believe are much more extensive and more compelling than shown by these other companies. We have placebo-controlled studies lasting many weeks. We have the dosing worked out. We have safety, and we have the mechanism of action of synaptic regeneration, all of which is only indirectly suggested by some of the data by Athira and Kasnova. Therefore, we urge you to consider the market cap ratio that we really deserve much more. This will allow us, even though we're fully funded, we have over $30 million in the bank. We believe we have a potential for a much higher market cap, which would give us the kind of bandwidth that would go after multiple targets and multiple ways of doing it at the same time. And for this reason, we urge you to consider the investment strategy with Synaptogenix in its, we believe, pioneering synaptic regeneration approach. In the next slide, briefly, our CEO, Dr. Alan Tuckman, is a superb neurologist and our chief medical officer, as well as our CEO, and myself, having been in the basic science and having joined several years ago full-time in Synaptogenix Incorporated. At this point, I'd be happy if there are any questions I might address.
There is a question here from Chris. Let's see here. Let me go to the chat here. Let me go to Chris Potter in the chat. Why do you think the phase 2 trial from a few years ago in Bryostatin failed or did not achieve statistical significance? Was it mainly because the people in the studies were taking the drug memantine? It's from Chris Potter.
Well, first of all, let me slightly modify that to say that it didn't fail.
Yeah, that was my words, not Chris's.
I think we were learning, Spencer. We were learning, and Cliff. We were learning the right conditions, and fortunately, we had pre-specified a cohort that did not have Namenda, which blocks the NMDA receptor. The NMDA receptor is very important for activity-dependent synaptogenesis.
Okay.
And PKC epsilon, activated by Bryostatin, regulates that receptor, the NMDA receptor, which is the target of Namenda. So by blocking it, we're blocking a major pathway. We had to learn that. So fortunately, we saw that in our pre-specified cohort, we saw, and I showed you some of that data, there wasn't a failure, it was a beautiful improvement. And I think this is somewhat distracting because people have to understand that as you learn, you understand what the real benefit is, and the real benefit was in the absence of standard of care Namenda, you get improvement over baseline. So, and we saw that same improvement in the second phase 2 trial, which we're now extending with a full 6-month trial that will actually read out 42 weeks after we start. But I urge you to look at some of the data, not only at AAIC, but in this paper we anticipate will be a peer-reviewed article shortly.
There's a couple more. Let's see if we can get to all these as we can.
Sure.
Let me go here. Has data from the last clinical trial been submitted for peer review on Bryostatin?
Yeah. The answer is yes. And all the data have been submitted. And we believe in all the pre-specified cohorts, which we believe will show a very compelling, statistically confident, placebo-controlled trial.
Should we expect the Fragile X trial to begin enrollment by the end of the year?
Yes. Well, I can't guarantee that. We're certainly working very hard to start it as soon as possible, maybe in the first couple months of next year. But we are working very closely with DuPont and Moore, which has superb collaborators. So we're working with going to the FDA, and the same thing with MS. We are doing that, and we still have a third trial, which we hope to announce shortly as well.
This is a question, I don't even know if I know what this, can you ask about the test subject? I don't even know if that's a question, but they're asking me if I can ask you about the test subject. I don't really know. I don't know what the question is there.
Well, I think if I'll look at it both ways. If people want to get enrolled, we have a channel by which you can certainly explore that. We welcome people to explore that. And I can tell you what the test subjects are they have many mental status exams of moderate to advanced disease. That means anywhere from as low as 18 all the way to as low as 10. That's our window. So we're looking at those types of patients.
Got it. Okay, yeah, we're at time. It is 3:08. Thank you so much, Dan Alkon, President and Chief Scientific Officer of Synaptogenix, ticker SNPX on the NASDAQ. And thanks so much for coming on the Benzinga Small Cap Healthcare Conference today.
It's my pleasure. Thanks for having me.
All right, I see our next